So this video is going to go through the process of mitosis. So when most people hear the word mitosis, they think of cell division. And yes, mitosis is a part of cell division, but technically, mitosis is nuclear division. So to get a little bit more specific, the whole point of it is to equally distribute all of the copied or duplicated DNA into two new nuclei. And the reason you want to do that is so that when the cell actually divides, both of your new cells are going to get an identical copy or set of DNA. We want to make sure that your new cells have the right amount of DNA in both cells. So let's look back. So you'll recognize we have the cell cycle drawn over here, and we know that growth happens during G1, then we have synthesis would be DNA replication, G2 a little bit more growth, and then M, we have mitosis, which we're going to look at, and then when the cell actually divides, that's cytokinesis. So if I were to just take a look at what my chromosomes look like during G1, by G1 I've got my DNA, or my chromosome, and by the time I am done, by the end of my S phase, my chromosomes have duplicated. So now I've got one set, and now I've got another set of DNA. So by the end of the S phase, which stands for DNA synthesis, we've duplicated or copied our DNA or our chromosomes in a sense. So if I look at my duplicated DNA, you're going to notice something called a centromere. And a centromere is this kind of red dot in the picture here, and it's a protein that holds these two sides or two kind of pieces of chromosomes together. These two identical pieces of chromosomes are called sister chromatids. So after DNA replication, if we were to coil our chromosomes, this is what they would look like underneath the microscope. So both sides, this side and this side, they are identical because that was the whole point of the S phase, is to duplicate your DNA, and those are called sister chromatids. So if I recap the cell cycle, I start with G1, I duplicate the DNA, we grow a little bit more time. This is all part of interphase. And then we enter mitosis, or the S phase. And the whole point here is I'm looking for nuclear division. So now I take my double DNA and I'm going to equally distribute it. So to just recap interphase, if I were to look underneath the microscope, our chromosomes are going to be uncoiled, which means they're not going to be visible underneath the microscope. So if I have my nucleus here and I were to look underneath the microscope, I really wouldn't see any chromosomes because they are uncoiled, so they're super thin strands, so I really wouldn't see them underneath the microscope. So if I look over in the picture here, here's an animal cell underneath the microscope, and I can tell because of its circular shape. Here's our nucleus, I'll outline that in black. And the chromosomes, I said, they're not really condensed, they're not very easy to see. So I can't see the chromosomes very well. During interphase, this is where the DNA is actually being copied. And that makes sense. I'm literally unwinding the helix in the first place, so I'm not really going to see this. So let's take a look at the very beginning of mitosis. So I'm starting to divide up my nucleus. So the first phase of mitosis is called prophase. And so a lot of different things happen during prophase. If I'm going to rearrange my nucleus, it would make sense that my nuclear membrane is going to break down. So that's why I'm representing them with dashed lines, showing that the nuclear membrane is breaking down. The second thing that's going to happen is now my chromosomes are actually going to become visible. They're going to condense and condense and coil. So now I'm going to start to see these chromosomes. Now, if this were an actual human cell, you would see 46 pairs of these, or 46 of these, 23 pairs. We don't have enough room to draw all this, so I'm just going to show 4 total chromosomes. So another thing that starts to happen is your microtubules begin to form these spindle fibers. So we start getting these spindle fibers that start to develop just during prophase. Next thing happens are centrioles, which are these red structures. They start to position themselves at the poles of the cell, or they move towards the ends of the cell. And our microtubules are going to be attached or anchored to the centrioles. And so the last part is you're going to see these microtubules here, they're going to kind of anchor themselves to the ends or to the sensors of these centrioles here. Okay, so that is most of what's happening during prophase, which is the first step of mitosis. Lots going on during prophase. And don't worry, not all the steps are this complicated.